Hi, this is Tutor Ivan Serunjoji. In this tutorial, we are looking at the introduction to carbon-4 plants or H-Slack pathway / C4 pathway. Introduction to C4 plants and overview. In this tutorial, we are going to be looking at basically 4 questions. Those answered, we shall be done. The first question shall be, why are they called C4 plants? The second question, where are they widely distributed? Then the third question, what are their examples? And then lastly, we shall look at the description of what we call the Kranz anatomy. This is a special anatomy possessed by these C4 plants. Introduction to C4 plants continued. Let's answer our first question. Why are they called C4 plants? Well, these are called C4 plants because these plants have their first stable product of photosynthesis as a 4-carbon compound. 4-carbon. So C in this place stands for carbon. Carbon-4 plants. And this is none other than oxaloacetic acid, or we can call it oxaloacetate. In short, OAA, all letters capital. Continuing with our introduction, the second question to be answered, having known the reason as to why these plants are referred to as C4 plants, we need to see where are they widely distributed on the continent of Africa and in the entire world. C4 plants are widely distributed in the tropics. Basically, that is in countries like India, Africa, some parts apart from North East Africa, there are some tropic areas. Well, our third question, what are their examples? We want to look at some of the examples of C4 plants. These include maize, in Lusoga, Duma, in Luganda, Kasoli, rice, in Luganda, Omuchere, wheat is also part of the C4 plants, millet, sorghum, and all other grasses. Basically, grasses are entirely C4 plants. Well, having looked at the examples, the distribution, and the reason as to why they are called C4 plants, we need to bear in mind that they have a certain pathway that leads to the first stable product being oxaloacetate, OSC4 plant. And this pathway was discovered by two scientists. These were agriculturists. That was Arch and Slack. Hence the name Arch-Slack pathway. Okay. Let us describe their special anatomy, or the Kranz anatomy. Basically, the word anatomy means the internal structures. So we want to look at the internal structures of these plants and why their internal structure is referred to as the Kranz anatomy. So, in short, when you are to describe, you have to tell us what you see in words. At times, you need to report to us with a drawing. We shall give you the pictorial of their internal structure or their anatomy, and this is called the Kranz anatomy. In brief, let us look at it. C4 plants have a leaf anatomy known as the Kranz anatomy, in which two groups of cells surround each vesicular bundle. As you see, the vesicular bundles, we have that the xylem and the phloem. The inner ring of cells is called the bundle sheath cells, and the outer ring is made up of the common mesophyll cells. So the bundle sheath cells are a special type of cells only found in the carbon-4 plants, OC4 plants. This arrangement of the bundle sheath cells around the vesicular bundles and the mesophyll cells around the bundle sheath cells is what we refer to as the Kranz anatomy. We shall see its significance as we shall look at the biochemistry of C4 plants in our next tutorial. So, the chloroplasts in the two rings are different morphologically. That is the outward appearance. Therefore, C4 plants contain dimorphic chloroplasts. Di meaning two. The word morphic coming from morphology. Two body forms, we can say. So when we look at the structure of the leaf of a C4 plant, internal structure, you see that the bundle sheath cells are a bit circular as opposed to the mesophyll palisade cells, which are cylindrical. So the morphology of these two cells, types of cells, is different, and therefore C4 plants possess, the morphology of these two types of organelles, the chloroplasts, is different. And being two, we have the word di, then due to the morphology, morphic. So they have, or they contain dimorphic chloroplasts. Having looked at that, you can find the transverse section of a leaf of a C4 plant showing how bundle sheath cells and mesophyll cells are arranged around the vesicular bundle. In short, the Kranz anatomy, in a textbook known as Functional Approach by Roberts, page 158. You can look at it and try to draw it. You draw it, then get used to it. So in that structure, you see the xylem and the phloem. Those are the vesicular bundles in the center, surrounded by the bundle sheath cells. Here we shall see that the Calvin cycle takes place here during the biochemistry of C4 plants. Then surrounded by another layer of palisade cells where we shall see that carbon fixation takes place. And those are the important chloroplasts we look at. Yes, we also have the spongy mesophyll cells surrounding, which give us the air spaces from which we extract carbon dioxide. So, thank you for watching. Endeavor to click the subscription button, that red button, in order to subscribe to this channel. In order to subscribe to this channel and not miss any of our tutorials. In our next tutorial, we shall look at the biochemistry of C4 photosynthesis, or the C4 pathway, or the arch-Slack pathway. Thank you for your time.